Welcome back to this next video in which we are discussing the visual system. Okay, so we've now discussed the way in which rod photoreceptor cells are going to pass the electrical signal onto rod bipolar cells and how they in turn are going to pass the electrical signal onto rod ganglion cells. What we now want to do is discuss the equivalent for cone cells, and this gets a little bit more complicated and we need to also discuss the trichromatic theory of light, how we actually see in colour. Okay, so let's begin by saying that actually the first bit is very similar. So if I bring back up my picture of a cone cell here, so if we have the cone cell synapsing onto a bipolar cell, which of course we will have, in response to hyperpolarization of the electrical potential difference across the cell membrane of the cone cell, you're going to get less glutamate released onto the cone bipolar cell. Now, this is where it gets more complicated. There are two different types of cone bipolar cell, whereas in the case of rod bipolar cells there were not two different types. So here's a new complexity. So with cone bipolar cells you have two different types. You have what are called on cone bipolar cells, and you have what are called off cone bipolar cells. And the on cone bipolar cells, these are the ones that are going to depolarize in response to the amount of glutamate going down, i.e. they're going to be activated when the cones have actually been activated by light. And these therefore need to have the same receptor for glutamate that we've seen on the rod bipolar cells. So they're going to have the mGlu-R6 receptor on the surface of them, just like the rod bipolar cells, and the exact same thing will happen for these on cone bipolar cells as happened for the rod bipolar cells, i.e. when the cones become activated by light, they hyperpolarize, they stop releasing glutamate, or they release less glutamate, and that stops activating these mGlu-R6 receptors, and that means that you're no longer hyperpolarizing the bipolar cell so much, so it starts releasing more glutamate onto the cone ganglion cell. In contrast, the off-cone bipolar cells, these are very different. So the off-cone bipolar cells, they have a more usual glutamate receptor. In particular, they have the AMPA receptor. I'll just move that over there. Okay, which if you like, you can call the AMPA-R. So this is the AMPA receptor. So this is a more normal receptor for glutamate. And if I just draw one of these terminals for a bipolar cell here, an AMPA receptor is a ionotropic glutamate receptor. It's a ligand-gated ion channel. So glutamate binds to the AMPA receptor, triggers the AMPA receptor to open, and then the AMPA receptor allows sodium ions to move into the cytoplasm of the cell, and therefore depolarizes the electrical potential difference across the cell membrane. And therefore this is going to have a very different effect, because these off-cone bipolar cells, when the glutamate is usually present, and remember the cones will be releasing most glutamate when they have not been activated by light, so when the cones are releasing glutamate, it'll be causing the AMPA receptors to open, and therefore you'll be depolarizing the electrical potential difference across this off-cone bipolar cell normally, and therefore the off-cone bipolar cell will be releasing more glutamate onto the cone ganglion cell, which will also be an off-cone ganglion cell, and therefore it will be firing action potentials very rapidly when there is no light present. Okay, so this is the complexity, that we now have these on-cone bipolar cells and these off-cone bipolar cells, and these will be synapsing onto ganglion cells, which we can then call on-ganglion cells and off-cone ganglion cells. So really, to make it utterly thorough description, we should put in cone here. Okay, so just to make sure this is absolutely clear, if we are talking about an off-cone bipolar cell, then when the cones that innervate that off-cone bipolar cell are activated by light, they will reduce the amount of glutamate they release. That means the AMPA receptors will start to close, and therefore you'll get more hyperpolarized electrical potential difference across the cell membrane. You'll stop depolarizing the electrical potential difference across the cell membrane, and therefore the off-cone bipolar cell will start releasing less glutamate onto its ganglion cell, therefore its ganglion cell will start firing electrical potential, it'll stop firing action potentials as frequently, and therefore we'll call it an off-cone ganglion cell. Okay, so this is a new complexity then, that with cone photoreceptor system, you can either have the activation of the cone represented by on-cone ganglion cells, which will start firing action potentials more rapidly in response to the cone being activated, or you can have it delivered to the brain by these off-cone ganglion cells, which will deliver the message to the brain by reducing the frequency at which they're firing action potentials. So there are these two different ways, basically, of delivering the information from certain cones on the eye to the brain, and in different parts of the retina, these two different options are used. Right, so let me now talk about the convergence of cone cells then. So, we've discussed how convergence of cone cells is not as big a deal as it is for the rod cells, and indeed at the fovea there isn't convergence, so each one of the cone cells in the fovea will have its own bipolar cell, which will be either an on bipolar cell or an off cone bipolar cell, and then each of those bipolar cells will have its own ganglion cell, and if it was an on cone bipolar cell, then it will have an on cone ganglion cell, and if it's an off cone bipolar cell, then we'd call its ganglion cell an off-cone ganglion cell. And just to make sure that this message is absolutely crystal clear, if we've got a cone that is going through the on system, then we will know that that cone has been activated by light if the ganglion cell is firing more rapid action potentials to the brain. Okay, so if this ganglion cell is firing action potentials more rapidly, that shows us that the cone photoreceptor was activated if it was wired through one of these on wiring systems, whereas if it was wired for an off wiring system, then the way that we'll know that that cone cell has been activated is by a reduction in the frequency of the action potentials that we're receiving. So it's just two different ways of coding the information, either for an increase in the frequency of action potential firing or for a decrease in the frequency of the firing of action potentials. So don't be afraid of this. It's just two different separate ways of encoding information, two opposite ways of encoding information, but the actual fact that a visual stimulus has hit a cone will still be delivered, just in these different ways. Okay, so different cones use these, at different points on the retina, use these different ways of conveying the message to the brain, but the brain can cope with this, basically. Okay, it knows which cones are wired up to on networks and which are wired up to off networks. So in the fovea we don't have any convergence. Outside of the fovea we do have convergence, but nowhere near as much as we had for the rod cells. And remember, when we do have convergence of cones, the cones that are converging onto a single bipolar cell need to be of the same type. So you can have lots of S cones all converging onto the same bipolar cell. In addition, we now have to add an additional complexity, because when we have the bipolar cells converging on a ganglion cell, so if I bring this picture up again, so with regards to the cones converging on the bipolar cell, they all need to be the same type of cone, and when we have bipolar cells converging on a ganglion cell, these bipolar cells need to be the same type of bipolar cell. So they need to be attached to the same type of cone cells, but also they need to be either all on cone bipolar cells or all off cone bipolar cells. So that adds an additional complexity. So you have to, each ganglion cell has to have all of the same type of cones feeding into it, and the bipolar cells need to all either be on bipolar cells or all off bipolar cells, because the on bipolar cells versus the off bipolar cells, they are encoding the information about activation in completely opposite ways. So you can't then mix them up when they're coming to a ganglion cell. You have to keep them separate so that these two different ways of signalling the activation of the cones remain fundamentally separate. Okay, right. So the cone system then, as we went through earlier, it has much lower visual acuity. Okay, no it doesn't. It has much higher visual acuity but much lower sensitivity. So its sensitivity is lower because the number of cone cells that are overall feeding into a ganglion cell is much lower than for the rod photoreceptor system, and therefore you require more intense light to get a ganglion cell to actually start conveying an electrical signal, whether it's an on ganglion cell, in which case the electrical signal will be an increase in firing rate of action potentials, or if it's an off ganglion cell, then the electrical signal will be a decrease in the frequency of firing action potentials. But it does have much higher visual acuity because of the same reason. Okay, so you can either have high sensitivity and low acuity, or you can have low sensitivity but high acuity, and the cone system is the high acuity one with low sensitivity. And this means that it's best under photopic conditions, and it doesn't saturate under photopic conditions in the same way that the rod photoreceptor system saturates. So under photopic conditions you are using your cone photoreceptor system, not your rod photoreceptor system. Okay, the final thing that we now need to discuss is the trichromatic theory of light. Now this is complicated, so we need to discuss how are you actually going to tell what colour light is arriving at a portion of the retina. And the theory of how this works is called the trichromatic theory. The trichromatic theory of colour, if you like. Okay, so let's say that on a little patch of retina we have got some green light hitting it. Okay, we want to know how are we actually going to tell that that is green light. Well, people's initial answer to this is, oh, well, it'll be because the green cones, the M cones, will be activated. They'll send signals to the brain, whether those are off signals or on signals, it doesn't matter, but they'll send signals to the brain, and that's the way that we'll know that it's green light, because it's coming from those green cones. But you have to remember what I told you. Green light doesn't just activate the green cones, it activates the other cones as well. It will activate the red cones and the blue cones, it's just it will activate those less well than it would activate the green cones. So the green cones will send more electrical activity, whether it's on signals or off signals, to the brain than the red cone system and the blue cone system. So what I'm trying to convey here is that it's not the absolute activation of the green cone system that tells you that the light is green. What you have to do is compare the amount of electrical signals that you are getting from all three types of cones, the S, M and L cones. And the way that you would work out that this is green light is you'll get most activation of the M cones, and you'll get smaller electrical signals coming from the S cone system and the L cone system. So it's by looking at the ratio of the electrical signals that you're getting from the three different cone systems that are pervading all over the retina. So you have these three different cone systems all over the retina. So by looking at the ratio of electrical signals that you are getting from these different types of cone system, that is how you can work out what the actual wavelength of the light that is hitting that part of the retina actually is. So that's the trichromatic theory, that it's the ratio of the electrical signals that you're receiving from the three different cone systems that allows you to actually tell what wavelength light it is. So a little test now to see if you've understood it. What would happen if you only had one type of cone cell? So let's say for some reason, maybe genetic reason, that you can't make the S-opsin and the L-opsin. Okay, so if you've got mutations in the genes that code for the S-opsin and the L-opsin, let's say, then you wouldn't be able to produce S cones or L cones, and you'd only be able to produce M cones. Would you be able to see in colour then? And the answer is absolutely no, you wouldn't be able to see in colour, because you wouldn't have the correct information. And you might say, but surely we would be able to see in colour, because if it's green light that's hitting that spot, then we'll get much more activation of these cones than if it's blue light. Okay, and that's a good argument. It's a good argument that we need to knock down. Okay, so let me repeat that argument. So you might think that you'd still be able to tell that this light is green, because surely it will result in much more activation, much more electrical signals coming from these M cones than you would have had if you'd shone the equivalent blue light on here. And I agree, if you shone equivalent intensity blue light onto that portion of the retina, you would get less activation of these M cones than if you shone green light. However, you could get the exact same electrical signal activity from the M cone system just by making the blue light more intense, and that's the key thing to understand. If I was to shine blue light onto this portion of the retina, but I was to make it far more intense than the green light that I had shone, so far more photons arriving of the blue light, then even though each blue photon has less chance of actually activating one of the M-iodopsin receptors inside the M cone, I'm still going to get the equivalent activation of the M cones that I would have got with the green light, because I've now shone more blue photons on, I've shone more intense light. So that's the problem here. You can't just do it with one type of cone, because you don't know whether this high activation due to the green light is because it is green light, or whether it's just another type of light but very intense, that sort of light. And that's why you can't do it with just one type of cone alone. This is known as monochromacy. Okay, so when you've just got one type of cone, this is called monochromacy, or more commonly, just colour blindness. Right, so that's the trichromatic theory of light. That's why you need these different types of cones, and actually you'd be able to do it just with two types of cone. Think through, you will hopefully agree that you'd be able to do it with just two different types of cone. You don't actually need 3 different types of cone, and I believe lots of other animals do just have 2 different types of cone cells rather than 3. Humans do, however, have 3. And it certainly does the job. I mean, there's no harm in having 3. It's just you don't actually need 3. You could do it just with 2. Okay, right, so there's the trichromatic theory of light, and there's how we're going to send the electrical information about cone activation to the brain, how we're going to send it to ganglion cells at the moment. Now what we need to do is follow how these ganglion cells are going to send their axons into the brain. So I now want to turn my attention to the central visual pathway. But before we do, I just want to clarify what we have spent so long talking about. What was the purpose of all of this bit that I've gone over about the rod photoreceptor system, the cone photoreceptor system? This was to make sure that you understand how we're actually going to encode the visual information in electrical signals. Okay, we have so far seen that rod information is going to be encoded in more frequent firing of those rod ganglion cells. We've seen that cone information can be encoded in either more frequent firing of ganglion cells, or it can actually be encoded in less frequent firing of action potentials by ganglion cells, depending on whether we're looking at an on circuit or an off circuit. Okay, that's a very important understanding, that cones can send this information to the brain either in these on signals or these off signals, but it does the same job of telling you where there is light present. Finally, we've looked at how the 3 different cone systems can together be used as part of this trichromatic theory to work out, for a certain portion of retina, what wavelength light is actually hitting there. And it's not the absolute activation of these different types of cone photoreceptor systems, it's the ratio of activation of the different ones, because the absolute activation doesn't just change with wavelength, it also changes with intensity. So you have to look at the ratio, not the absolute value of how much electrical activity is coming or is changing. Right, so that's what we have discussed now. So I hope what I have convinced you is that we have now successfully encoded information about intensity of light that is hitting the different parts of the retina, and also wavelength of light that is hitting the different parts of the retina in this electrical activity, in these electrical signals. And now all we need to do is send these electrical signals to the brain, and in principle, if we send them to the brain could interpret them and work out something about the visual image that we are seeing and give us sight. So that's where I hope we're at the moment, an understanding that the eye has successfully encoded visual information into electrical signals, and now all we're going to do is very, very simple. This is the deep physiology part of it. This is the difficult bit to understand. All we're now going to have a look at is the anatomy part of it. How is this information going to be transmitted to a portion of the brain that can then analyse this information? So the first thing that I want to do is see how the axons from these ganglion cells are then going to get out of the eye. So the axons are all going to head towards the optic disc, which is where the optic nerve begins, and they're all going to go into the optic nerve of their eye. Now, the axons that are running in the fibre layer of the retina, they are not myelinated, and they're not going to become myelinated until they actually get into the optic nerve. Right, so let me draw this here. So we'll begin with a little drawing here. So we'll have the two eyes. So we'll have, here is our right eye here. So it's just a very simple picture. So I'm just showing the right retina really rather than the entire eye. And here, this can represent the left retina. So what's going to happen is that each of them is going to send all of the axons from all of the ganglion cells, and maybe you can remember the number that I gave you for how many ganglion cells there are in each eye. Remember, there is 1 million ganglion cells per eye, so all those 1 million ganglion cells all over the retina, they're going to send their axons into the optic nerves, which are what I'm showing here. So these are the optic nerves, also known, of course, as the second cranial nerves. Okay, and once the fibres get into the second cranial nerves, they're then going to become myelinated. Now, an important point here that I would just like to stress is that the cells which are going to myelinate the axons of the ganglion cells in the optic nerves are not Schwann cells. They are in fact oligodendrocytes. So Schwann cells are the cells which myelinate axons in the peripheral nervous system. Oligodendrocytes are the cells which myelinate axons in the central nervous system. So I'll write down these names. So Schwann cells are in the peripheral nervous system, and oligodendrocytes are in the central nervous system. So the big important thing to understand here is that these are effectively not part of the peripheral nervous system. The optic nerve, you can view it really as part of the central nervous system rather than as a peripheral nerve, because the axons in there are not myelinated by Schwann cells, but instead are myelinated by oligodendrocytes. Okay, so I'll stress that again. The axons of the ganglion cells here are going to be myelinated by oligodendrocytes rather than Schwann cells. Now, what is actually the difference between a Schwann cell and an oligodendrocyte? Well, a Schwann cell tends to just myelinate one axon, so this is the way a Schwann cell would work. So if we have the axon here that's going to be myelinated, okay, and it's just a little section of the axon, of course it would have bits on either end, it wouldn't just end here and here, okay, but this is just a little portion of the axon that we're interested in. What would happen is a Schwann cell would just myelinate one neuron's axon. So the way that myelination works is that the cell effectively flattens out. So I'll try and draw this like so. So here is this Schwann cell. Here is its nucleus up here. Right, and what effectively you do is you stretch the cell. So it's a cell like any other cell. It's got a cell membrane with cytoplasm, and what you do is you can imagine stretching the cell and sort of rolling it out like, I don't know, pastry maybe, with a rolling pin. Imagine rolling the cell out so that you've got this sort of flat portion of the cell where you've got hardly any cytoplasm in between the membrane, the two sides of the cell membrane. And then what you just do is you wrap this sort of cell round and round the neurons. So you'd wrap this forward here, and then you'd continue on. So you'd bring this bit up here, and you'd wrap it round and round and round, and what you end up with is like a Swiss roll. So here's the axon, and then here is our Schwann cell wrapping round and round, and I've sort of messed it up a little bit there, so let me try and fix it. Okay, so it would wrap round and round and round like this, but you'd always have each of these lines. It's not just one phospholipid bilayer, it's two phospholipid bilayers effectively put together. So I think I'll try actually drawing this picture again. So here's the axon, okay, So the axon there at the centre, and then this can represent the cytoplasm of the cell, and basically the cell is going to be wrapped around and around and around, but then get rid of these spaces, wrap it really tightly. That's what myelin actually is. It's these cells wrapped round and round and round axons, and of course that means that you get layer after layer after layer of cell membrane basically, and that's the lipid insulation of axons. Okay, so why was I even drawn into this? Yes, the difference between Schwann cells and oligodendrocytes is a Schwann cell will only do this for a single axon, whereas oligodendrocytes are quite brilliant. They do it for more than one axon. So an oligodendrocyte might look like this. Here is its central cell body, and then what it has coming off is loads of little processes like this, and then these processes will expand and then do this wrapping around, so it will expand out like this, and here's the bit that you can wrap around a neuron. So imagine putting an axon here and then wrapping this round and round that axon, and then again you can have the same thing coming off over here. You can put an axon through there and then wrap this round and round the axon to produce its myelin. So that's the difference between Schwann cells and oligodendrocytes. Schwann cells just go around a single axon, whereas oligodendrocytes, they have a central cell body with lots of processes coming off, and they then have these produce myelin that will surround loads of axons rather than just one. And the difference between myelin and just normal cell membrane is that you do put slightly different lipids in this portion of your cell membrane. So when a cell is going to make a myelin sheath, so when a Schwann cell is going to make a myelin sheath or a myelin sheath, it will change the consistency of this cell membrane compared to the cell membrane that you'd have over at the cell body. So it is a strange cell membrane basically. It's different sort of lipids in that membrane to the normal composition. Okay, and that's why myelin is special, and we don't just call it a normal cell membrane lipid composition, because it's not a normal cell membrane lipid composition. It has loads of different lipids that you wouldn't find in a normal cell membrane. So why have I stressed this point so much, that the optic nerve, unlike all the other nerves that you might think of as being part of the peripheral nervous system, is not actually part of the peripheral nervous system, because it has its axons myelinated by oligodendrocytes rather than Schwann cells. Why have I stressed this point so much? Well, the reason I've stressed this is because there is a disease called multiple sclerosis, which I'm sure you will have heard of, often abbreviated down to MS. And multiple sclerosis is a autoimmune condition where the oligodendrocytes are attacked by the immune system. So the immune system attacks by mistake the oligodendrocytes, which myelinate cells of the central nervous system. And one of the first classic presentations of multiple sclerosis is optic neuritis, inflammation of the optic nerve, and it's because the immune system has attacked the oligodendrocytes that are myelinating the axons of the ganglion cells in the optic nerve. So a little clinical correlate there. So one of the first presentations of MS can be optic neuritis, and the symptoms that you'd get if you suffered from optic neuritis is you'd get sudden damage to your vision. So either you'd get complete blindness in one eye, or you'd get reduced vision in that eye, so all your vision would go blurry. Okay, and that's because the vision is clearly being disturbed for that eye that is inflamed. And you'd also get pain of the eye, particularly when moving the eye. So that's optic neuritis, and it is one of the classic first presentations of multiple sclerosis, and it occurs because the immune system has attacked the oligodendrocytes within the optic nerve, and therefore it's important to know that in the optic nerve you have oligodendrocytes rather than Schwann cells, because Schwann cells are not attacked by the autoimmune reaction of multiple sclerosis. Okay, right, so the myelination begins in the optic nerves here, and I will finish this video by just drawing on the optic chiasm there, but I will stop here, and in the next video we'll take it from here and we'll start the central visual pathways. We'll start by discussing the optic chiasm, then we'll discuss the optic tracts, and we'll go on to discuss how this visual information is going to be used. So we'll discuss the pupillary light reflex, how it's going to get to the primary visual cortex, and then finally we'll discuss what lesions to different portions of the central visual pathway will actually result in.